3, 2, 1.
Good morning, and thank you for the opportunity of sharing some opening thoughts this morning. So understanding the organization of cells and tissues and how this organization influences function is a fundamental quest in the life science research. Over the years, technological advances have shed much light on these aspects of biology. Sequencing approaches have allowed us to learn about cell types and their heterogeneity, and methodological advances in microscopy have transformed our understanding of cells and tissue structure and functions. Spatial genomics, or more in general, spatial biology, to include proteins too, or also called spatial multiomics, are changing the way we understand complex tissue and cell interactions. The flexibility to apply these technologies to both hypothesis-driven and discovery-driven studies allows widespread applicability in research settings. For this reason, you probably know already, the method of the year 2020, selected by Nature, is the spatially resolved transcriptome. And of course, at Nanostring, we're very proud to be part of such important choice. The broad utility of the spatial approach is reflected in the array of strategies that have been developed for its implementation, as seen in the recent commercial technologies development. We saw them yesterday in day one of this interesting conference. We also heard and commented yesterday that there are many open questions around how do we understand and compare all these new technologies and solutions? How do we understand what works well for what experimental design and what is actually destined not to maybe never really take off? So to try and help you answering these questions in the spatial revolution, first of all, I like to bring to your attention one very recent technology review with title Applicability of Spatial Transcriptional Profiling to Cancer Research. This is a recent publication published in Molecular Cell, and it's reporting the comparative work carried out from UCSC, from the Troy Ishron lab team. This review compares methods across many technology, as well as highlighting applications that can best benefit from spatial analysis work. So I encourage you to have a look at this sort of publication, but also I would like to share some personal thoughts on why Geomics DSP can help you in your work, in your spatial work. And I'm going to share some high-level information that I hope it helps you clarify how we can help. So one of the methods that allows one to study the transcriptome in space can be described generally as spatially resolved RNA sequencing or spatial sequencing. There are multiple commercial solutions and even experimental homebrews. Here, the concept is that basically at a high level, you have an array which is placed on your tissue sample in order to capture and retrieve the messenger RNA out from the tissue into the array, often with a poly-A capture. You read out this positionally captured transcriptome information with sequencing that gives you the high throughput, very desirable. These assays come at various spatial resolution, but as of today, it seems that what they have in common is that they provide multicellular information because the arrays consist of regularly spaced grids that do not completely adapt to the cell's organization profile. The alternative to this method has been mostly in the realm of microscopy with high multiplex RNA imaging, which can give good resolution in a tissue. Most examples are, frankly, at a lower plex, but for higher plex imaging, there are specialized microscopy setups that are required, and multiple cycles of imaging are needed too. So at Nanostring, we thought from the start that the combination of spatial sequencing and microscopy provides a great solution to measure RNA spatially with Geomics. So Geomics does offer cellular resolution for cell types, which is different from single cell resolution. Come at the booth if you want to understand this better. Geomics does use probes against selected genes panel or even against the full transcriptome with up to 22,000 genes, the entire whole transcriptome for human and mouse. Geomics does use sequencing as a readout, giving you the high throughput, and it does allow you to measure RNA and protein. On top, the instrument has been really developed to work on archival FFPE tissue, which is a very good advantage. Because you're not drawing the RNA molecules out of the tissue with a fixed array, with Geomics, you can follow the tissue contour and select specifically or exclusively, for example, the cancer tissue, or maybe just focus exclusively on another tissue type. Maybe you want to only see the stromal content, so you can adapt your choice and selection and segmentation. So there are already more than 40, actually 48 publications underpinned by research using Geomics that are a testimony to the success of this novel platform. I will post in the chat in a moment the link to this paper. Finally, and without promising the future too much, know that if you have different research questions that do require subcellular resolution of high-plex panels, you heard already yesterday from our Chief Technology Officer, we are launching the Spatial Molecular Imager, and we are accepting technology access projects. Come to the booth, virtual booth, if you want to know more. To close, if you would like to hear more about how Nanostring can support your spatial work, you can listen to our presentation from yesterday morning or afternoon. You can meet us at the booth, and I look forward to meet you in the next occasion in person at the live conference. Finally, tomorrow, there is actually another session, the convolution sessions, to look more into the bioinformatics of this great technology on spatial, looking at spatial biology. So don't miss the session with Patrick Danhart. Have a great day, and thank you for listening.